The genetic code. At the very heart of the central dogma is the concept of information transfer from the linear sequence of the four-letter alphabet of the polynucleotide chain into the 20 amino acid language of the polypeptide chain. As we have seen, the translation of genetic information into amino acid sequence takes place on ribosomes and is mediated by special adapter molecules known as transfer RNAs or tRNAs. These tRNAs recognize groups of three consecutive nucleotides known as codons with four possible nucleotides at each position. The total number of permutations of these triplets is 64, like 4 into 4 into 4 value, well in excess of a number of amino acids. Which of these triplet codons are responsible for specifying which amino acids and what are the rules that govern their use? In this chapter, we discuss the nature and underlying logic of the genetic code, how the code was cracked, and the effect of mutations on the coding capacity of messenger RNA. The code is degenerate. One of the most striking features of the code is that 61 of the 64 possible triplets specify an amino acid, with the remaining 3 triplets being chain terminating signals. This means that many amino acids are specified by more than one codon, a phenomenon called degeneracy. Codons specifying the same amino acids are synonyms. For example, UUU and UUC are synonyms for phenylalanine, whereas serine is encoded by the synonyms UCU, UCC, UCA, UCG, AGU, and AGC. In fact, when the first two nucleotides are identical, the third nucleotide can be either cytosine or uracil, and the codon will still code for the same amino acid. Often, adenine and guanine are similarly interchangeable. However, not all degeneracy is based on equivalence of the first two nucleotides. Leucine, for example, is coded by UUA and UUG, as well as by CUU, CUC, CUA, and CUG. Codon degeneracy, especially the frequent third place equivalence of cytosine and uracil or guanine and adenine, explains how there can be great variation in the AT or GC ratios in the DNA of various organisms without correspondingly large changes in the relative positions or proportions of amino acid in their proteins. For example, genomes of certain bacteria display vastly different AT-GC ratios and yet more closely related enough to encode proteins of highly similar amino acid sequences. Then perceiving order and the makeup of the code. Inspection of the distribution of codons in the genetic code suggests that the code evolved in such a way as to minimize the deleterious effects of mutations. For instance, mutations in the first position of a codon will often give a similar, if not the same, amino acid. Furthermore, codons with pyridines in the second position specify mostly hydrophobic amino acids, whereas those with purines in the second position correspond mostly to polar amino acids. Hence, because transitions AT to GC or GC to AT substitutions are the most common type of point mutations, a change in the second position of a codon will usually replace one amino acid with a very similar one. Finally, if a codon suffers a transition mutation in the third position, rarely will a different amino acid be specified. Even a transversion mutation in this position will have no consequence about half the time. Another consistency noticeable is the code is that whenever the first two positions of a codon are both occupied by G or C, each of the four nucleotides in the third position specifies the same amino acids such as proline, alanine, arginine, and glycine. On the other hand, whenever the first two positions of the codons are both occupied by A or U, the identity of the third nucleotide does make a difference. Since G to C base pairs are stronger than A to U base pairs, mismatches in pairing, the third codon base are often tolerated if the first two positions make strong GC base pairs. Thus, having all four nucleotides in the third position specify the same amino acid may have evolved as a safety mechanism to minimize errors in the reading of such codons. Then, Wobble in the anticodon. It was first proposed that a specific tRNA anticodon would exist for every codon. If that were the case, at least 61 different tRNAs, possibly with an additional 3 for the chain terminating codons would be present. Evidence began to appear, however, that highly purified tRNA species of known sequence could recognize several different codons. Cases were also discovered in which an anticodon base was not one of the four regular ones, but a fifth base, eosin. Like all the other minor tRNA bases, eosin arises through enzymatic modifications of the base present in an otherwise completed tRNA chain. The base from which it is derived is an alanine whose carbon 6 is deaminated to give the 6-keto group of eosin. Eosin is actually a nucleoside composed of ribose and the base hypoxanthine, but it has come to be referred to as a base in common usage, and we do so here. In 1966, Francis Crick devised the Vobel hypothesis to explain these observations. It states that the base at the 5' end of the anticodon is not as specially confined as the other two, allowing it to form hydrogen bonds with any of several bases located at the 3' end of a codon. Not all combinations are possible, with pairing restricted to those shown. For example, U at the Vobel position can pair up either adenine or guanine, while I can pair with U, G, U, C, and A. Pyrimidine pairs would give ribose distance that are too long or too short, respectively. The Vobel rules do not permit any single tRNA molecule to recognize four different codons. Three codons can be recognized only when eosin occupies the first 5' position of the anticodon. Almost all the evidence gathered since 1966 supports the Vobel concept. For example, the concept correctly predicted that at least three tRNAs exist for the six serine codons UCU, UCC, UCA, UCG, AGU, and AGC. The other two amino acids like leucine and arginine that are encoded by 6 codons also have different tRNAs for the sets of codons that differ in the first or second position. In the three-dimensional structure of tRNA, the three anticodon bases as well as the three following 3' bases in the anticodon loop all point in roughly the same direction, with their exact conformations largely determined by stacking interactions between the flat surfaces of bases. Thus, the first 5' anticodon base is at the end of the stack and is perhaps less restricted in its movements than the other two anticodon bases. Hence, Vobel in the third position of the codon. By contrast, not only does the third 3' anticodon base pairing in the middle of the stack, but the adjacent base is always a bulky modified purine residue. Thus, restrictions of its movement may explain why Vobel is not seen in the first 5' position of the codon. 3 codons direct chain termination. As we have seen, 3 codons do not correspond to any amino acid. Instead, they signify chain termination. As we discussed in chapter 11, this chain terminating codons UAA, UAG, and UGA are read not by special tRNAs but by specific proteins known as release factors RF1 and RF2 in bacteria and eRF1 in eukaryotes. Release factors enter the A site of the ribosome and trigger hydrolysis of the phenyl peptidyl tRNA occupying the P site, resulting in the release of the newly synthesized protein. How the code was cracked. The assignment of amino acids to specific codons is one of the great achievements in the history of molecular biology. And how are these assignments made? By 1960, the general outline of how messenger RNA or mRNA participate in protein synthesis has been established. Nevertheless, there was still little optimism that we would soon have a detailed understanding of the genetic code itself. It was believed that identification of the codons for a given amino acid would require exact knowledge of both the nucleotide sequence of a gene and the corresponding amino acid order in its protein product. At that time, the elucidation of the amino acid sequence of a protein, although a laborious process, was already a very practical one. On the other hand, the then current methods for determining DNA sequences were very primitive. Fortunately, this apparent book did not hold up. Roadblock did not hold up progress. In 1961, just 1 year after the discovery of mRNA, the use of artificial messenger RNAs and the availability of cell-free systems for carrying out protein synthesis began to make it possible to crack the code. Then stimulation of amino acid incorporation by synthetic mRNAs. Biochemists found that extracts prepared from cells of Escherichia coli that were actively engaged in protein synthesis were capable of incorporating radioactively labeled amino acids into proteins. Protein synthesis in these extracts proceeded rapidly for several minutes and then gradually came into a stop. During this interval, there was a corresponding loss of mRNA owing to the action of degenerative enzymes present in the extract. However, the addition of fresh mRNA to extracts that had stopped making protein caused an immediate resumption of synthesis. Then, the dependence of cell extracts on externally added mRNA provided an opportunity to elucidate the nature of the code using synthetic polyribonucleotides. The synthetic templates were created using the enzyme polynucleotide phosphorylase, which catalyzed the reaction HMP-N plus XDP gives XMP N plus 1 plus P, where X represents the base and XMP raised to N represents RNA of the length N nucleotides. Polynucleotide phosphorylase is normally responsible for breaking down RNA and under physiological conditions favors the degradation of RNA into nucleoside diphosphatase. By use of high nucleoside diphosphatase concentration, however, this enzyme can be made to catalyze the formation of internucleotide 3' 5' phosphodiester bonds and thus make the RNA molecules. No template RNA or DNA is required for RNA synthesis with this enzyme. The base composition of the synthetic product depends entirely on the ratio of various ribonucleotide monophosphates added to the reaction mixture. For example, when only adenosine monophosphate is used, the resulting RNA contains only adenylate acid and this is called polyadenylate acid or poly A. It is likewise possible to make poly U, poly C and poly G. Addition of the two or more different diphosphate produces mixed copolymers such as poly AU, poly AC, poly CU and poly AgCu. In all these mixed polymers, the base sequences are approximately random, with the nearest neighbor frequencies determined solely by the relative concentrations of the reactants. For example, poly-AU molecules with 2 times as much A as U have sequences like this. Then poly-U codes for polyphenylalanine. Under the right conditions in vitro, almost all synthetic polymers will attach to ribosomes and function as templates. Luckily, high concentrations of magnesium were used in the early experiments. A high magnesium concentration circumvents the need for initiation factors and the special initiator F-MET tRNA, allowing chain initiation to take place without the proper signals in the mRNA. Poly-U was the first synthetic polyribonucleotide discovered to have mRNA activity. It selects polyphenylalanine tRNA molecules exclusively, thereby forming a polypeptide chain containing only phenylalanine. Polyphenylalanine. This we know that a codon for polyphenylalanine is composed of a group of 3 uridylic acid residues UUU. That a codon has 3 nucleotides was known from genetic experiments, and on the basis of analogous experiments with poly C and poly A, CCC was assigned as a proline codon and AAA as a lysine codon. Unfortunately, this type of experiment did not tell us what amino acid GGG specifies. The guanine residues in poly G firmly hydrogen bound to each other and from multi-stranded triple helixes that do not bind to ribosomes. Then mixed copolymers allowed additional codon assignments. Poly-AC molecules can contain 8 different codons like CCC, CCA, CAC, ACC, CAA, ACA, AAC, and AAA, whose properties vary with the copolymer AC ratio. When AC copolymers attach to ribosomes, they cause the incorporation of asparagine, glutamine, histidine, and threonine, in addition to the proline previously assigned to CCC codons and the lysine previously assigned to AAA codons. The proportions of these amino acids incorporated into polypeptide products depends on the AC ratio. Since the AC copolymer containing much more A than C promotes the incorporation of many more asparagine than histidine residues, we conclude that asparagine is coded by 2AS and 1C and that histidine is coded by 2CS and 1A. Similar experiments with other copolymers allowed several additional assignments. Such experiments, however, did not reveal the order of the different nucleotides within a codon. There is no way of knowing from random copolymers whether the histidine codon containing 2 Cs and 1 A is ordered CCA, CAC, and ACC. Then transfer RNA binding to defined trinucleotide codons. A direct way of ordering the nucleotides within some of the codons was developed in 1964. This method utilized the fact that even in the absence of all the factors required for protein synthesis, specific amino acyl tRNA molecules can bind to ribosome mRNA complexes. For example, when poly U is mixed with ribosomes, only phenylalyl tRNA will attach. Correspondingly, poly C promotes the binding of prolyl tRNA. Most importantly, the specific binding does not demand the presence of long mRNA molecules. In fact, the binding of trinucleotide to a ribosome is sufficient. The addition of the trinucleotide UUU results in polyalanine tRNA and attachment, whereas if AAA is added, lysyl tRNA specifically binds to ribosome. The discovery of this trinucleotide effect provided a relatively easy way of determining the order of nucleotides within many codons. For example, the trinucleotide 5' GUU 3' promotes valine tRNA binding, 5' UGU 3' stimulates cysteine tRNA binding, and 5' UUG 3' causes leucyl tRNA binding. Although all 64 possible trinucleotides were synthesized with the hope of definitely assigning the order of every codon, not all codons were determined in this way. Some trinucleotides bind to ribosomes much less efficiently than UUU or GUU, making it impossible to know whether they code for specific amino acids. Then codon assignments from repeating copolymers. At the same time, when the trinucleotide binding technique became available, organic, chemical, and enzymatic techniques were being used to compare synthetic polyribonucleotides with non-repeating sequences. Ribosomes start protein synthesis at random points along these regular copolymers, yet they incorporate specific amino acids into polypeptides. For example, the repeating sequence CU is the messenger for a regular polypeptide in which leucine and serine alternate. Similarly, GUG promotes synthesis of a polypeptide containing two amino acids, cysteine and valine, and ACAC directs the synthesis of a polypeptide alternating threonine and histidine. The copolymer built up from a repetition of the three nucleotide sequence AAG directs synthesis of three types of polypeptides: polylysine, polyarginine, and polyglutamic acid. Poly-AUG, AUC behaves in the same way, acting as a template for polyisoleucine, polyserine, and polyhistidine. Further codon assignments were obtained from repeating tetranucleotide sequences. The sum of all these observations permitted the assignments of specific amino acids to 61 out of the possible 64 codons, with the remaining 3 chain terminating codons like UAG, UAA, and UGA, not specifying any amino acid. Then, 3 rules governing genetic code. The genetic code is subject to 3 rules that govern the arrangement of and use of codons in messenger RNA. The first rule holds that codons are read in a 5' to 3' direction. This is a principle, and as an example, the coding sequence for the dipeptide NH ARGCOH would be written as 5 prime ACG CGA 3 prime, where 5 prime ACG 3 prime is a threonine codon and 5 prime CGA 3 prime and arginine codon, or as 3 prime GCAAGC 5 prime, wherein the codons are written in the same order. The second rule is that codons are non-overlapping and the message contains no gaps. This means that successive codons are represented by adjacent trinucleotides in register. Thus, the coding sequence for the tripeptide NH2-THR-ARGCOH is represented by 3 contiguous and non-overlapping triplets in the sequence 5 prime ACG, like that. The final rule is that message is translated into a fixed reading frame which is set by the initiation codon. Translation starts at an initiation codon which is located at the 5 prime end of the protein coding sequence. Because codons are non-overlapping and consistent of 3 consecutive nucleotides, a stretch of nucleotides could be translated in principle in any of 3 reading frames. It is the initiation codon that dictates which one of the 3 possible reading frames is used. Thus, for example, the sequence given could be translated as a series of threonine codons and series of arginine codons like the picture, and depending on the frame of the substream, start codon. Then 3 kinds of point mutations alter the genetic code. Now that we have considered the nature of the genetic code, it is instructive to revisit the issue of how the coding sequence of a gene is altered by point mutations. An alteration that changes a codon specific for one amino acid to a codon specific for another amino acid is called missense mutation. As a consequence, a gene bearing a missense mutation produces a protein product in which a single amino acid has been substituted for another, as in the classic example of a human genetic disease sickle cell anemia, in which glutamate 6 in the beta globin subunit of hemoglobin has been replaced with a valine. A more drastic effect results from an alteration causing a change to a chain termination codon, which is known as a nonsense or stop mutation. When a nonsense mutation arises in the middle of genetic message, an incomplete polypeptide is released from the ribosome owing to premature chain termination. The size of the incomplete polypeptide chain depends on the location of the nonsense mutation. Mutations occurring near the beginning of a gene result in very short polypeptide, whereas mutations near the end produce polypeptide chains of almost normal length, as we saw, and mRNAs that contain a premature stop codon are rapidly degraded in eukaryotic cells by a process known as nonsense mediated mRNA decay. The third kind of point mutation is a frame shift mutation. Frame shift mutations are insertions or deletions of one or a small number of base pairs that alter the reading frame. Consider a tandem repeat of the sequence GCU in a frame that would be read as a series of alanine codons. The codons are artificially set apart from each other by a gap for clarity, but are of course contiguous in a real messenger RNA. Now imagine the insertion of A in the message, thereby generating a serine codon AGC at the site of the insertion. The resulting frame shift causes triplets downstream of the insertion to be read as cysteines. Thus, the insertion, or for that matter, the deletion of a single base drastically alters the coding capacity of the message, not only at the site of insertion but for the remainder of the messenger as well. Likewise, the insertion or deletion of 2 bases would have the effect of throwing the entire coding sequence at and downstream of insertions into a different reading frame. Finally, consider the instructive case of the insertion of 3 extra bases at nearby positions in a message. It is obviously that the stretch of message at and between the 3 insertions will be drastically altered, but because the code is read in units of 3, mRNA downstream of the 3 inserted bases will be in its proper reading frame and hence completely unaltered. Then next topic, genetic proof that the code is read in units of 3. The preceding example is a logic of classic experiment of Francis Crick, Sidney Brenner and They are co-workers involving bacteriophage T4 that established that code is read in units of 3 and did so purely on the basis of genetic argument without any biochemical or molecular evidence. Genetic crosses were carried out to create a mutant phage harboring three inferred single base pair insertion mutations at nearby positions in a single gene. Of course, the three insertions would have scrambled a short stretch of codons, but the protein encoded by the gene in question, called R2, was able to tolerate the local alteration to its amino acid sequence. This finding indicated that the overall coding capacity of the gene had been chiefly left unaltered despite the presence of three mutations, each of which alone or any two of which alone would have drastically altered the reading frame of the gene's message and rendered its protein production active. Because the gene could tolerate three insertions but not one or two, or for that matter, four, the genetic code must be read in units of three. Next, suppressor mutations can reside in the same or a different gene. Often, the effects of harmful mutations can be reversed by a second genetic change. Some of the subsequent mutations are easy to understand, being simple reverse back mutations, which change an altered nucleotide sequence back to its original arrangement. More difficult to understand are the mutations occurring at different locations on the chromosomes that suppress the change due to mutations at site A by producing an additional genetic change at site B. Such suppressor mutations fall into two main categories: those occurring within the same gene as the original mutation, but at a different site in its gene, intragenic suppression, and those occurring in another gene, intragenic suppression. Genes that cause suppression of mutations in other genes are called suppressor genes. Both of the types of suppression that we are considering here work by causing the production of good or practically good copies of the protein made inactive by the original harmful mutation. For example, if the first mutation caused by the production of inactive copies of one of the enzymes involved in making arginine, then the suppressor mutation allows arginine to be made by restoring the synthesis of the good copies of this same enzyme. However, the mechanisms by which intergenic and intragenic suppressor mutations cause the resumption of the analysis of good proteins are completely different. As an example of intragenic suppression, consider the case of a misgene mutation. Its effect can sometimes be reversed through an additional missense mutation in the same gene. In such cases, the original loss of enzymatic activity is due to an altered three-dimensional configuration resulting from the presence of an incorrect amino acid in the encoded protein sequence. A second missense mutation in the same gene can bring back biological activity if it somehow restores the original configuration around the functional part of the molecule. Then, next, intragenic suppression involves mutant tRNAs. Suppressor genes do not act by changing the nucleotide sequence of a mutant gene. Instead, they change the way the mRNA template is read. One of the best known examples of suppressor mutations are mutant tRNA genes that suppress the effects of nonsense mutations in protein coding genes, but mutant tRNAs that suppress missense mutations and even frame shift mutations are also known. In E. coli, suppressor genes are known for each of three stop codons. They act by reading a stop codon as if it were a signal for a specific amino acid. There are, for example, three well-characterized genes that suppress the UAG codon. One suppressor gene inserts serine, another glutamine, and a third tyrosine at the nonsense position. In each of the three UAG suppressor mutants, the anticodon of a tRNA species specific for one of these amino acids has been altered. For example, the tyrosine suppressor arises by a mutation within a tRNA TYR gene that changes the anticodon from GUA 3' AUG 5' to CUA, thereby enabling it to recognize UAG codons. The serine and alanine glutamine suppressor tRNA also arise by single base changes in their anticodons. The discovery that cells with nonsense suppressors contain mutationally altered tRNAs raised the question of how their codons corresponding to these tRNAs could continue to read normally. In the case of tyrosine UAG suppressor, the answer comes from the genes coding for tRNA TYR. One codes for major tRNA TYR species, whereas the other two are duplicate genes coding for species present in smaller amounts. One of the other of the two duplicate genes is always the site of suppressor mutation. No such dilemma exists for UGA suppression, which is mediated by a mutual form of tRNA TRP, and suppressing tRNA TRP retains its capacity to read UAG tryptophan codons while also recognizing UGA stop codons. This is possible because the anticodon was changed from CCA in the wild type to UCA in the mutant tRNA TRP, and verbal rules, as we have seen, allow recognition of A or G in the 3' position of the codon by U in the 5' position of an anticodon.